DNA damage occurs at a tremendous rate every day. 10 to the third to 10 to the sixth base pair changes occur. The largest impact is on base structure. However, you can get breaks between bases, double-stranded breaks of the DNA, a variety of modifications that impact the function of bases. As a consequence, cell survival depends on being able to repair the DNA, because any change in base pair can affect the function of the protein that's encoded by a segment of DNA. And while alterations in function can be advantageous, for example, a bacteria acquires antibiotic resistance by a change in an enzyme, they can also be deleterious. So you can render an enzyme completely non-functional by a single base pair change that results in its inability to assume its folded functional structure. So it's not surprising that a significant percentage of the coding capacity in all organisms is for repair enzymes, and that's on the order of 3 to 5% across the board, because DNA damage to a bacterial cell can be lethal because it's the only cell, whereas our cells can manage to have a cell or a few that's damaged as long as it doesn't have other untoward changes. So how does DNA damage occur? Well, our own metabolism generates oxygen radicals that can damage DNA bases. UV light damages DNA bases. It cross-links bases so that it makes it impossible to replicate the DNA. Ionizing radiation of various sorts creates DNA damage. Chemicals can modify DNA and alter its structure. And finally, replication errors can introduce mispaired bases. And all those forms of DNA damage can have impact on the generation of the proteins that are needed for cellular survival. But we also have a lot of ways to deal with DNA damage. In higher organisms, because of the cell cycle in eukaryotic cells, there is a moment where if DNA damage is present, it can be detected, and the cell cycle can be stopped to give the DNA repair systems time to do their business. You can also have transcriptional activation of specific proteins that are able to deal with different kinds of DNA damage. And on the far right, in the worst case, you can go to self-imposed cell death, called apoptosis or apoptosis. But there are also multiple mechanisms for directly repairing DNA damage. Direct reversal of damage, base excision repair, cut out the base, recopy it, make sure it's okay, nucleotide excision repair, mismatch repair, double-stranded break repair, called homologous recombination. So the cell is poised to respond to all of these different types of DNA damage, but because there's so many, it takes a lot of different proteins. And those proteins are critical because cells that do not or cannot repair DNA accumulate damage. One event happens, another event happens, and another. And cumulative damage can have combinatorial impacts on cellular function and survival, so that cells can become dormant, they can die or commit apoptosis, where the cell destroys itself, or perhaps in the worst case, particularly for eukaryotes, which have multiple cells, a single cell can be transformed. That leads to unregulated cell division, meaning cancer, and other types of diseases. So DNA repair is critical for the well-being of the cell and ultimately the well-being of the organism. The double-stranded structure of DNA, which lends itself to the repair process, simply uses the complementary strand for making the correction. It's not the same base, but it's the complementary base. So, as discovered when the structure was identified for DNA, you can read one strand's sequence by looking at the other strand. They're not the same, but they're complementary. So DNA repair encompasses a wide range of process. First of all, You have to identify the type of DNA damage in order to introduce the corrections that restore the DNA to its original structure. And there has to be, especially in eukaryotes, ways to signal other cellular systems when the repair is simply not possible to complete. That is, you want to say, okay, time to shut down the cell. And it's perhaps also not surprising that the rate of repair will vary by cell type, by cell age, by environmental conditions. There are a lot of signaling inputs into the repair process. So again, why is DNA repair so important? Well, in part, multiple human diseases that are inherited come because of repair defects that result in altered base pair changes. In addition, the central role of the repair process is reflected in the delay of the eukaryotic cell cycle in order to accommodate repair processes, in order to accommodate the completion of restoring the DNA to its original state. So, would you expect DNA repair to be more efficient in your grandmother than in your sister? Why? Why not? Can you identify ways to avoid the common types of risks for DNA damage? Sun exposure is the most obvious, but radiation, chemicals, other ways that DNA can get damaged.